Hi, I would like to tell you about illustrating viruses and how computer graphics can help us with it. But perhaps even more important question that I'll try to answer in this talk is, why would anybody want to do that? And I'll start very egoistically by telling you how did I end up making pictures of viruses. I studied at TU Wien, where I did a PhD in scientific visualization. And then together with colleagues of mine, we founded a startup where we are doing these visualizations commercially. But now I have to backtrack a little bit and tell you about procedural graphics. This is something that fascinated me since I was a teenager. And this is what it is. It's creating pictures with programming code. Here this is this famous example of a fractal, which is created by 4 simple transformations, which are expressed by just a couple of simple formulas. And the way this works is that we put a point on the screen, we draw it, and then we randomly choose one of these transformations and move the point to the new place where we draw it again and just repeat for many, many times. And this simple algorithm ends up with this very complex picture. And what I liked about this was that whenever I wanted to change something about this picture, I could just change my code and I would get a result. So for instance, if I wanted to see the distributions of these points, I could just make them transparent by changing these 2 numbers. And I would see the structure of this picture much better. And also all these numbers that you see there, it doesn't matter what they are doing, but if I change them, it will also change the picture. And so if I would decide to smoothly or continuously change these numbers, I would get an animation. And so this was very interesting for me, and I was playing a lot with these things. It especially works great with these self-repeating fractal type of pictures, which can be expressed by very simple rules, and they can create very complex images. And by changing a small detail, I can very easily introduce randomness, or I can introduce order. And so after some time, when I was playing with these things, I started to create worlds out of mathematical functions. And this was very interesting for me. This somehow, to me, reflected the beauty of mathematics, which would be expressed in a visual way. But after all, I wasn't a philosopher searching for beauty. I was a programmer. So I was creating these tools that would help me create such worlds. So for instance, I made this generator for textures, where one can take several operations and connect them in this node link diagram, which is now very common in this 3D modeling software or other applications. And here in this case, it was creating these textures out of these nodes, which were defining some simple operations and some simple mathematical operations and connections between them. And what was interesting in this case, though, was that there was this function where I could take this network, which is describing my texture, and I could generate a programming code out of it. So this was in Pascal, which is this old programming language that was created for teaching programming. And I was using it back in the day. And then I had this piece of the code that I could use in my programs to create these textures there and create this or use them in my world. So I was creating this very naive animations, but what was interesting was that everything here was described by mathematical functions. So what was kind of cool about it was that this would be then all saved in a small executable, which would be just several kilobytes in size, and I could change every aspect of this. And so later I was starting to experimenting more with this 3D and with different forms of lighting and rendering. And at some point I came to trying to mimic reality with these mathematical functions. So something that you could describe as a abstract realism, where you try to make functions that somehow are reminiscent of something that we know from the real world, like how materials behave, or how some objects would interact together. And so this would then later prove to be very useful for me in my work when I started to explore the field of molecular visualization. And to introduce this field to you, I will show you this picture. It's a it's a watercolor painting by David Kutzel of a E. coli bacteria cut in half so you can see inside. And you can see its membrane, its outer membrane in green with all this tail sticking out, and then in the middle you can see this yellow hairball. It's a genetic information, it's a DNA which is packed like this in the bacteria, and then the purple part are the protein molecules, so large molecules that are the basic building blocks of all cells, bacteria or viruses. And David Kutzel painted this picture in order to show us how crowded these environments are, how all these protein molecules that are essentially carrying out all the functions of the bacteria, they are encoding the life. Everything that the cell or the bacterium does is encoded in these interactions between these proteins. And here we can see how tightly packed they are, how full of molecules this bacterium is. And you can imagine that creating such a picture is very difficult. Here's a photograph of David painting one of his artworks, and you can imagine that creating such a artwork takes a lot of talent, a lot of patience, and of course a lot of biological knowledge. I mean, David is a biologist in the end, and so he knows what needs to be painted, and he also has the ability to do so. But this is extremely time-consuming process and cannot be recreated by just anybody. And so we were working with him and also with his team to create a software which would be using computer graphics to generate such pictures inspired by his visual style. But besides creating an interesting image, the question is, why would anybody need something like this? Why do we need molecular visualization? There are three areas where molecular visualization could be interesting or useful. And one of them is analysis. When biologists are studying diseases or trying to come up with new drugs that are curing these diseases, it's ultimately the question of how the molecules interact. It's a question of shapes, whether some small molecule will fit in some opening in the bigger one so that it can bind in a special position in there, so that this drug or this medication can work. These things like shapes of molecules, we can find them out, we can study them, but in order to create or to find out whether some molecule would fit with another one, visualization is a great tool which can help scientists in achieving that. And so it is analytical tasks, visualization is very helpful. Another one is modeling, where we are creating new materials, where the properties of the material would be dictated by the interactions of its molecule, and then creating models of such materials in virtual world with help of visualization can help us to make these materials better. And then the last area, which is most important or interesting for me, is storytelling. And imagine that I would like to tell you about bacteriophage. I would tell you that it's this weird shape with this hull and the tail with legs, and then just flies through the space until it hits the bacteria, and then it attaches to its membrane, and then it punches through, and it squeezes its genetic material inside, and then it tricks the bacteria to instead of using its machinery to create the proteins that it needs, to start creating the copies of the new virus and creating thousands of new viruses, which are then ultimately killing the bacteria and ripping it apart and flying off until they find another one. You know, it's interesting to hear about it, but in order to understand it, it would be much easier if you could actually see it, if I could show you what I mean by these things. And these processes, these things, they happen on a scale that is beyond the resolution of the most powerful microscopes. And so the only way to show you would be through illustration, animation, or visualization. And so there was one example that I was working on. It was about microtubules. There are these tubes that are inside of cells, and they are somehow holding the shape of the cell, so they are forming a sort of skeleton. but they are also used for transporting things, materials or signals from one side of the cell to the other. And what's interesting about them, they are very dynamic. They are breaking apart all the time and then they are being recreated. And so we wanted to model this, process of how microtubule is disintegrated and how it's formed again. It's something we cannot see in a microscope, because even with an electron microscope where we can see the microtubules, we have to freeze it, so we cannot see it moving. And so there are some theories that biologists have formed of how these microtubules are behaving, and so we try to model this. In order to do that, we designed this framework for creating these biological animations. I can explain on this simple example of a sphere. We would like to make an animation of this sphere made of protein molecules being formed from these randomly scattered molecules. And so easy way to do that would be to simply interpolate the positions of the molecules with the final positions on the sphere. And then we had a nice animation of a sphere forming. But this is not how things work in biology. There is specific order in which these molecules are being assembled. And this is what we want to capture. And so in our framework, we considered these initial and the final stage not to be some static keyframes, but functions, where we can procedurally describe the movements and the behaviors of molecules at the beginning and at the end. And then the interpolation would also take into account the specific paths that these molecules have to take in order to recreate some process. And so we would end up with something like this. We would have a framework which allows us to create these animations of biological processes. And with microtubules, it looked like this. So here's the microtubule when it's being broken down. This event is called a catastrophe, and biologists have a theory of how it looks like. So we model it here with our procedural approach. And then this breaks down the microtubule along until it stops at some point, and this is called a rescue, when the microtubule starts to be formed again by forming this sheet, which is then closed down, forming the tube. But I promised I will be talking about viruses. So let's start with the question, what does a virus look like? Well, here's a picture from an electron microscope which shows several particles of coronavirus. We can see their shapes. We can see these famous spike proteins that are projecting outside of its membrane. And yeah, so that's it. And we can even color individual parts of the virus so that we can see better how is it structured. We can scan this even layer by layer, so to move this, to create this moving cutting plane kind of visualization to get a better idea. This we can use to reconstruct the 3D model. We can even rotate it, look at it from different sides to get kind of an idea of what does a virus look like. We can, with this 3D model, cut through it so that we can see inside of it, see what's going on there. But even with this fancy 3D visualizations, we still don't quite get the idea. What does a virus look like? It's still very fuzzy, it's still very uncertain, not very tangible. The thing is, a virus is smaller than the wavelength of light, wavelength of the visible light. So a short answer to the question, what does a virus look like, is that it doesn't. We can never see it with our eyes, we can never see the light reflected of it, and so arguably it doesn't look like anything. We can use these instruments like electron microscopes to give us kind of an idea of its shape. Of course, it does have a shape, but to see any more details, the only way is to use molecular visualization. We know a lot more about the viruses than what we can see in the microscope. We know which proteins are forming it, how are they organized inside, and so we can fill the gaps in the pictures obtained from the electron microscopes with this knowledge that we have from various other experiments and from all this biological research to create a 3D model of a virus which is so detailed that we can zoom in all the way to see individual molecules and the atoms that are forming them. So we know that if we want to take a close look at a virus, we have to illustrate it. So what are the things that we have to consider when we are creating illustrations of viruses? First thing is the level of abstraction. since these are illustrations, we are the ones who are choosing how many details do we put in. And for the given purpose, we shouldn't try to put more details than that are necessary. Let's consider this illustration of a HIV virus. So if I would be telling you that HIV is kind of like round particle with these glycoproteins attached to its surface, and then inside there is this thing called capsid, which holds the genetic information in the form of RNA, which is shown in purple. And I would be telling you that maybe HIV has this reverse transcriptase, which is this enzyme that can actually turn the RNA into DNA, which is important when the virus replicates. So if I would be telling you these things, this illustration would be completely sufficient because everything that I said is already included there. And any additional details will be just distracting from this narrative that I wanted to convey. We can take a look at this illustration of the same virus particle. It's more detailed. Here we can see individual protein molecules. This is kind of a drawing or similar style to what David Goodsell would paint. And this kind of illustration is telling something else. It tells us how tightly packed these protein molecules are, how crowded this environment is. It shows us several important types of molecules with their distinctive shapes, but it's still not quite realistic. These molecules are way larger than they should be so that we can actually see the shapes. There are too few of them because otherwise they wouldn't fit in this picture. So we had to abstract these details in order to tell the information that we wanted to say, in this case of this crowdiness of the environment. And then we can go one step further. We can consider a full atomistic model of HIV particle where we would have individual models of individual protein molecules in atomic resolution, in actual size, with correct concentrations. Such an illustration would be important or would be useful when we want to communicate things like molecular processes. How does this virus work? How does it infect the cell? How does it replicate? All these things, they are the question of molecular processes that are happening inside of it, and explaining these things would be simplified by having such a visualization. Another thing is, with this advantage of having a model that is this detailed, is that we can create all the abstracted versions from it. So if we have a model with enough details, we can always simplify it. And so that's why these molecular models are important or interesting for us, because we know that if we want to communicate something that would require less details, we can simply remove details from our model. The other way around doesn't work, because in the abstracted model, the details are simply not there. And so this is why we are always working with these molecular models. Now, another thing to consider when visualizing viruses is scale. Viruses are small, we know that, but in order to convey this scale, we have to think of how to show the virus. We can typically put it next to something else, like a red blood cell, which, by the way, should look more like this, because on this small scale of red blood cell, the color doesn't work like it does in everyday objects. Nothing can be so vividly colored when it's so small, but I will talk about color a little bit later. Now, for this talk, I prepared several visualizations of viruses, and I will show it to you now. And each of these visualizations, they're all created with computer graphics, and they are all communicating some message. So let's take a look. So this is the first visualization of a virus. The large thing that you see, that's actually a human hair. The virus is just 1 pixel in this red circle. Despite the virus being shown as a single pixel in this image, it already tells you an important information, and it is how large the virus is. I could tell you that the virus is 100 nanometers across, but that wouldn't tell you much, because it's very hard to imagine for us. I could place it next to the red blood cell like I did on the illustration before, but this is also not so helpful, because it's not graspable to us how small or how large a red blood cell is. But everybody knows how thick a hair is. So if I put it next to a hair, it immediately tells you that this virus is really tiny and you can actually imagine it. I could tell you that the virus particle is 1,000 times smaller than the width of a hair. But it's not as striking as if you actually see it in a picture. Now, this is an illustration which shows the environment in which virus exists. So this is the epithelium, this lining of these tubes that go into your lungs, where you breathe, and some of them have this hair which are pushing things that shouldn't be there outside when you are coughing or sneezing. And the viruses are infecting these cells. In this case, the coronavirus is infecting these cells, and then you still see these viruses as these bright dots. You don't see much of them, but you can see the environment in which they are living. You can see this landscape in which they exist. A lot of visualizations or illustrations of viruses don't convey this. You often see illustrations of a cell as a kind of a sphere floating around, and the virus is a smaller sphere and it enters it and so on. But that's not how it is. These cells, they are forming tissues, so they are forming these landscapes, and this is the environment where the virus exists. And, these virus particles, they are just moving around. They cannot move themselves, so they are just pushed by all the other molecules that are here. So they are moving through Brownian motion, and when they hit one of these cells, they attach to it. The cell swallows them, and the virus then tricks the cell into creating new copies of this virus. And when one virus particle would enter such a cell, 1,000 new particles would exit. And so this is what we see in this picture, this 1,000 virus particle exiting a dead cell that was previously infected. And so all these newly created virus particles will be then maybe blending on some of the surrounding cells, infecting those. Here we have another visualization where we actually are looking at the detail of the surface of a cell. Here we can see these shining dots. They are the virus particles attached to the surface of the cell. This kind of visualization shows us the invasive nature of the virus, how they are covering all these folds and how they are everywhere, how they are difficult to get rid of because they are not existing in one place. They are kind of everywhere. When they are spreading so fast in our organism, they would be just found in all these dark alleys of surfaces of the cells. And so such a visualization, again, doesn't really show us the virus, but it shows us where it exists, so that we can imagine kind of the life cycle of it, not just this one particle. And of course, we can also make a visualization just like this one, where we can see the individual particle of a virus. This kind of visualization is also helpful when we are communicating maybe how does the virus work. When we want to say that the virus have these spike proteins, and these spike proteins are actually attaching, they are chemically bonding with receptors which are on the surface of some of our cells. And when this binding happens, then the virus is sucked inside a cell where it infects it. And when we want to say something like this, such a molecular model where we can see individual molecules which are doing these things is very helpful. And then we can also combine all of these scales together in one picture, because the virus, it's not just this particle, which is biologists call it virion. Virus is not only the virion, it's not this little sphere that we always see in all the pictures. That's not the virus, that's just one of its forms. The virus is a complex agent. It lives inside of the infected cells, where it tricks the cell, it reprograms its machinery so that the cell, instead of producing the things that normally it would produce, it starts producing parts of new virus particles. And these are then assembled. Then the virus is ripping its membrane apart and wrapping itself in it, and then it exits the cell, and it's pushed around and tries to infect something else. So this, when it's in this form of a virion outside of the cell, it's not living. It's just a dead particle which is pushed around. And all of these things, this whole life cycle of a virus, when we want to visualize it, we really have to consider several scales. So combining them in one picture is one way, but the problem would be that it's difficult to see what these things are. I mean, on this particular picture, we see this couple of virions from the left side kind of like being pushed towards the cell. In the foreground, we see one particle which already attached to a receptor, and so will be later swallowed. Then further on the right, we see one of these virions being sucked in, where the membranes of the virion and the cell merge together, and all the insides of the particle are spit into the cell where they do their work. And then afterwards, way in the background on top right, we can see many new virions exiting the cell that was previously infected. And so this picture kind of illustrates this life process, but the scale is not very well conveyed, because to fit these scales that are so many orders of magnitude apart, we have to use very strongly forced perspective. And so we could mark maybe things, how large they are in the foreground, in the background, to kind of give us this idea of the perspective. Or maybe we can do something like this: zoom in a part of the image in the background, show these little virus particles, and then show that one of these particles is actually the one that we see in the foreground. So to kind of explain this scale, which when we are talking about processes like a life cycle of the virus, this is inherently multiscale. And so another thing to consider besides level of abstraction and scale is the color. As I kind of mentioned before, virus doesn't have a color. It's smaller than something that could have a color that is visible. So when we are creating these visualizations, we have to assign colors. We have to choose which colors do we use. For this particular visualization of coronavirus, we wanted to use the color scheme to communicate something. So not to be just arbitrary, but to really communicate that the virus isn't a living thing. So that's why we chose this kind of overall cold color scheme. But also it does contain RNA, which is this genetic information that can that causes that the virus can be replicated inside of a cell. And so this genetic information is arguably the most living part of the virus, and that's why we showed it in bright red, this kind of like active thing. And also these spike proteins on the outside, we show them in this vivid pink to kind of signify that these are the parts of the virus that are actually doing something. They are all the parts are doing something, but these are the ones who are actually attaching to the cell and causing the virus to be swallowed by the cell. And they are also important they are important target for the medications, because they can be recognized from the outside. And the other parts, like for instance this membrane around the virus, that membrane is actually coming from the cell that was previously infected. The virus, when it's formed, it rips part of the membrane of the cell and it wraps itself in it. So this part is arguably dead. It's a part of a virus that doesn't do anything besides holding everything together. And so that's why we chose this cold color for it. Same with these proteins which are holding the RNA together. They are not living things. They are just structural kind of scaffolding for the RNA to hold it together. So that's why we made them blue as a kind of like cold color. Of course, as I said, this choice of colors is arbitrary, and so sometimes we choose color based purely on aesthetics to make the picture pretty. But no matter what color scheme do we use, the important thing to realize is that these visualizations are multiscale, and so should be the color scheme. Like in this example, we color the virus according to its parts, so each part is different color. But when we zoom in to see individual molecules, we see all the molecules and all the atoms of the same color. It doesn't tell us much. This is kind of useless. And so when we are looking at it at this scale, we could actually color the atoms. But then when we zoom out again and we have the colors of like all the atoms colored in different colors, we only see noise, because at this scale, color scheme which distinguishes individual atoms doesn't make sense. And so this is why colleagues of mine have developed this method which takes arbitrary color scheme and it assigns it to the model. But it assigns it not based on all the data inside of the model, but based on what you actually see right now. So when you're up close, you can see the atoms colored differently, but when you zoom out, you can see the individual compartments of this model colored differently. And no matter what you see, the color scheme is assigned to all the objects so that it's utilized in the most efficient way. These changes are always continuous, so that it never looks like the colors are jumping around or changing abruptly. So you can still understand what is this that this is one coherent model. And so this was a method developed for exploration of, interactive exploration of these large multi-scale data sets. The message here is that no matter what color scheme do we choose, we have to consider the scale, and we have to merge these colors and scale together so that they are dynamically assigned and based on what we actually see in our visualization. I mean, with things like viruses, where you can see entire virus particle, which is 100 nanometers across, and then you can see individual parts of it, like the capsid, or you can see the molecules that are forming these structures, or you can see even the atoms which are forming the molecules. These are scales which are orders of magnitudes apart, and so this has to be considered when assigning the colors. So we have all these methods for visualizing viruses, but in order to visualize something, we need to have the model. And these models are very complex, so it's difficult to create them by hand. And so in our work, we are using these procedural approaches. Our gateway to this world of molecular visualization was a project with our collaborators from the Scripps Institute. They developed the software called CellPack, which is able to take atomistic models of molecules and fill predefined compartments with them. And they use this software to create the model of HIV particle. So you can see in a microscope how this particle looks like, what's the shape, what are the shapes of the compartments. And from some other experiments, you can find out which kind of protein molecule should be inside. And so they used the CellPack to create a model of this. And it was a large model, and it took several minutes to render. And so they asked us if we could come up with something that could render it a little bit faster. And so we created a software which is able to take this model and visualize it in real time. And this gave us new possibilities, because now the users could interactively explore the model, could look inside and see what's going on in there. And this wasn't possible before, because this model was simply too huge to be practically used. The problem is that this model is still a static one. And so it reflects knowledge or state of knowledge at some point. But whenever this knowledge changes, that biologists learn something new about the virus, in order to integrate it in this model, it would have to be recreated from scratch. So the scripts for the CellPack would have to be changed, then the CellPack algorithm would have to be run again, which can take hours, and then we would have a new version of the model. But if we would like to visually tweak it by changing some parameters and immediately see how the resulting model looks like, so that we can more closely match our knowledge about it, this will be impossible. And so we decided to go one step further, and we decided to generate these models in real time while they are being rendered. So we came up with this method which can also fill spaces and compartments with molecules, but it only does so where the camera is looking. So it splits the space into regions, and only those regions which are currently visible would be filled with molecules in real time. So this could give us this possibility to interactively tweak the parameters and see how the model is changing, so that we can actually create something that we know how it should look like from the experiments, but we don't know how to describe this easily in these parameters. We don't know what these parameters should be, so we have to try them out and see how the results look like. And so this novel way of creating these molecular environments in real time gave us this possibility. And we also did this for generating genomes, so this DNA or RNA molecules, which are very long molecules created from these several bases which are linked together in a long chain. And we made this generator which can create this in real time. So we can create these long molecules in our models without having to do this manually, or without having to wait for a long time while it's generated with a CellPack. And we made it scalable, so we can create this DNA for viruses, but also for bacteria, which are way larger than viruses. So here we can see example of three bacteria, and the size of the DNA molecule that would be inside of them. So these are huge models which are then generated automatically, and our method is then able to visualize them in real time. And we did this also for the generator of lipid bilayers. So lipid bilayers, they are membranes which are enclosing cells and viruses, and they are made of two sheets of lipid molecules, and essentially what they are doing, they are keeping the fluid inside and the fluid outside separate. So whenever we want to create a model of a bacteria or virus which has this membrane, we can use this generator. It works in a way that it takes an arbitrary mesh and it populates it with these two sheets of lipid molecules without repetitions, without seams, and does so interactively. So we can visualize these models of these membranes also in real time, which is important for us. And so we have now these three components that can generate models of proteins. So these large molecules, lipids, which are these membranes, and genetic information, so the DNA or RNA. And these are the basic building blocks of all viruses and bacteria. And so this gives us ability to create models of any virus if we know how it's supposed to be organized. And so there was this lockdown in spring, and so I had I was spending a lot of time at home, and so I decided I'll try to model this coronavirus. So I knew there are these three types of proteins on the membrane, so I created the membrane and populated it with these three types of protein. And then inside I knew there is another protein, so I filled the space with this algorithm with this type of protein, and then use this generator for the RNA to create this RNA strand with a random walk. And so I had this very rough model of how coronavirus could look like from the outside and from the inside. But it was back when this research was ongoing, right? So there were some of these models of these protein molecules, they were not available yet. So I took the ones from the original SARS, because it's likely that they are going to be very similar with in a new one. And so I created this virus with these procedural methods, which also means that whenever there will be a new model of this protein released, which is what these biologists are doing all the time, I could simply replace it in my model without any additional effort. And that's very important, because I knew that this is just a preliminary model and it's going to change once there is a paper coming out which tells me that this protein actually looks a little bit different, or there are many more of them or less. So I can modify my model easily. But that's exactly the thing. The ideal situation would be that this knowledge about these viruses could be integrated in such models directly by biologists. Because if I want to do this, I have to change my scripts and I have to do the programming. So it would be great to have some sort of environment where programming is not required at all. And we can create very easily these models, and so that we can give this environment to the biologists and they can create these models themselves. And this is especially important in times like this, where there is a new paper about this particular virus coming up every day, pretty much, and this knowledge changes very quickly. And to keep our model up to date, the only way is that the biologists would be creating these models themselves. And so that's why my colleagues from KAUST came up with this pipeline of creating these molecular models with a statistical and rule-based system. And I will guide you through this pipeline very quickly to give you an idea how this works. And so it starts with a segmentation step. We start with a microscopic picture of this virus that we want to model. And in this picture we have to segment the shapes. So we have to outline the shapes so that the system can learn what are the representative shapes of these viruses. And we also mark the proteins that are projected from the outside of the membrane, so that the system can learn how are they distributed on the membranes in a typical case. So we do this for several virus particles, and then from now on the system is automatic. So it is based on machine learning models. And what it does, it takes all these contours of the shapes of the virus particles, and it takes these distributions of these proteins and sends them to the next stage, in which these contours are processed with a machine learning model, and a representative contour is created from them. And so there is a 2D mesh created out of this contour, which kind of tells us what's the typical shape of a virus particle. This shape is then taken into the next stage, where it's inflated into a 3D mesh. So now we have a 3D mesh which is representative of how a virus would look like in 3D. And these distributions of these proteins that were on the membrane that we marked in the first stage is also projected on the surface of this 3D mesh. So now we know where it's more likely that these proteins on the surface would be positioned, and the system can do this automatically. So it automatically fills the membrane with the correct amount of these proteins and in the correct distributions, so that it's somehow representative of our viruses that we saw in the microscopic image. And then again comes the part where the user has to interact with the system, and it is the part where the rest of the model is created by showing several examples to the system. So we would place several molecules that we know are inside of this virus, and we would position them in a way how they should be positioned. And every time we do something like this, the system is learning the rules and tries to fill up the rest of the model with this given set of rules. So here's the example of how RNA strand could be created by placing these bases next to each other. So we know that, or the system knows that these type of molecules are kind of touching or connecting together, forming long lines. And then we would twist one of them, and the other ones would be twisting in the same way. And the system would learn to understand that we want to create such a spiral strand. So this gives us ability to very rapidly model complex models, such as those of viruses, and then in the last stage we can just render them. And so there is no programming involved. It's really just looking at microscopic images and knowing what they mean and marking it for the system, letting the system to process it, and then giving it several examples of how this model should be constructed. And so in this way, the biologists themselves can create these models of this level of complexity. And with this system, we created the model of a coronavirus which reflects the latest research. So this is the latest stage of our knowledge that we have about how the virus looks like from the outside and from the inside. And with this model, we then created the following video. Our goal is not to just have a system for modeling viruses. We would like to have an environment for sharing knowledge. So an environment where biologists could enter all the knowledge that they learn about things that they study, and the system would prepare all the necessary models and visualizations that could be deployed anywhere, could be used as figures for the papers that the scientists are publishing, or to create an interactive application for lay audiences to learn about these things and understand them better. And so all the different styles and all the different levels of abstractions that need to be incorporated in these visualizations or illustrations would be provided by the system, and they would be created automatically based on the input from the biologists, from the illustrators, from whomever is using this environment. And so all this research, all this work that we do is somehow heading towards this direction of having such an environment at some point. And so that's why we try to deploy our visualizations to various environments. We try to deploy this to the VR so that the users can explore a virus firsthand. They can dive inside and see what's going on there. We also deployed it to large displays, to a planetarium. So here you can see a large dome, which with my colleague down there for scale. And so this is a planetarium for 100 people where they all can be immersed in this complex molecular environment, which is the great tool for learning about complex things like this. And so my talk was entitled "How to Illustrate a Virus," and so I would like to end with showing you a short animation that I made that somehow fast forwards through the process of how we created our latest illustration of the coronavirus. Thank you for your attention.